Black walnut. In yards across the eastern United States, gardeners spend years trying to grow tomatoes, roses, or blueberries near a black walnut tree. The plants keep dying. Pests, disease, and watering are all ruled out. The tree is the problem. And it has been poisoning the soil the entire time. Juglone nigra, the black walnut, is native to the central and eastern United States and is one of the most commercially valuable hardwood trees in North America. Its nuts are edible. Its lumber commands premium prices, and its roots, leaves, bark, and husks continuously release a compound called juglone into the surrounding soil. Juglone is a phenolic compound that interferes with the enzymatic processes plants use to convert oxygen into usable energy. When a susceptible plant absorbs it through the roots, cellular respiration slows and eventually fails. The symptoms look exactly like drought stress: yellowing leaves, wilting, stunted growth, because the plant is functionally suffocating at the cellular level even in well-watered soil. Tomatoes, apples, blueberries, rhododendrons, pines, and many other common garden species are highly sensitive. Grasses, maples, and certain others tolerate it. The zone of active toxicity extends 50 to 60 feet from the trunk in a mature tree, often well beyond the visible drip line. Fallen leaves and decomposing husks add to the soil concentration each autumn. The compound persists after the tree is removed. Gardeners who cut down a black walnut and replant the area frequently find the soil remains inhospitable for 3 to 5 years, sometimes longer, as juglone breaks down slowly in the presence of soil bacteria. The Penn State and Illinois extensions both document consistent reports of this pattern from gardeners across the eastern states. Black walnut matters because it is one of the most common trees in American backyards. And the damage it causes to neighboring plants is invisible, continuous, and outlasts the tree itself. Garlic mustard. This plant does not outcompete the forest understory by growing faster or taller. It destroys the underground system that the forest depends on to function at all. Alyria petiolata, garlic mustard, is a biennial herb native to Europe and Asia that has spread across the eastern United States and southern Canada since its introduction in the 1860s. It grows in forest understories, along trail edges, and in shaded woodland gardens. It is recognizable by its triangular leaves, small white spring flowers, and the sharp garlic odor released when the leaves are crushed. Garlic mustard releases a compound called sinigrin, a glucosinolate also found in broccoli and Brussels sprouts, into the surrounding soil. Sinigrin disrupts mycorrhizal fungi, the underground fungal networks that form symbiotic relationships with tree roots and help them absorb water, phosphorus, and nitrogen from the soil. When garlic mustard invades a forest stand, the mycorrhizal network that connects and supports the trees begins to fail. Peer-reviewed studies have documented reductions in mycorrhizal colonization of competing plant species by over 50% in garlic mustard-infested soils. Nitrogen availability dropped by more than 50% and phosphorus by approximately 17% in affected areas. The effect is most damaging to tree seedlings, which depend on mycorrhizal connections during early establishment. Native tree regeneration slows significantly in forests where garlic mustard has persisted across multiple growing seasons. Research also shows that allelopathic effects persist in the soil after the plant is removed, suppressing native recovery even when garlic mustard is no longer physically present. Garlic mustard matters because it wins not by being stronger than its competitors, but by dismantling the biological infrastructure those competitors depend on to survive. Tree of Heaven. This tree arrived in Philadelphia in 1784 as an ornamental import. It now grows in every contiguous U.S. state. Part of why large-scale removal has never succeeded is that it poisons the ground before any competitor can take its place. Ailanthus altissima produces a compound called ailanthone in its roots, bark, leaves, and decomposing leaf litter. Ailanthone is one of the most potent natural herbicides documented in plant research. It suppresses germination and early growth of competing species by disrupting cell division and photosynthetic function. In controlled studies using sterilized soil, ailanthone proved more effective at suppressing radish growth than several commercial herbicides tested in parallel. A single tree produces over 300,000 seeds per year, each capable of wind dispersal across significant distances. But the allelopathic chemistry the root system maintains in surrounding soil means that even when competitors germinate nearby, the chemical environment inhibits their establishment. The tree poisons the competition before the competition becomes a problem. The U.S. Forest Service lists Tree of Heaven as one of the most ecologically damaging invasive species in the country. Effective removal requires immediate herbicide treatment of cut stumps to prevent regrowth from the root system. Without that step, the root system persists underground, continues releasing ailanthone into surrounding soil, and produces rapid surface regrowth from multiple points simultaneously. The visible portion is removed while the underground chemical factory continues operating. Tree of Heaven matters because it combines prolific seed production, rapid growth, and active soil poisoning into a single system where each mechanism reinforces the others. Spotted knapweed, in the rangeland and grasslands of the American West. This plant has converted millions of acres of diverse native prairie into near monocultures. It does not achieve this through physical competition alone. Centaurea stobe, spotted knapweed, is native to Eastern Europe and has spread across over 7 million acres in the western United States, primarily in Montana, Idaho, Oregon, Washington, and Colorado. It is one of the most economically damaging invasive plants on western rangelands, reducing forage production for livestock and displacing native grasses and forbs that support wildlife across large areas. Spotted knapweed releases catechin, a polyphenolic compound, from its roots into surrounding soil. Catechin triggers a reactive oxygen cascade in the root cells of neighboring grass species, effectively inducing programmed cell death in competing root systems. Native North American grasses have no evolutionary history with catechin and no resistance mechanisms against it. European grasses, which evolved alongside knapweed on its native continent, have developed resistance. The North American native species encounter a compound their biology was never exposed to before. Research at the University of Montana confirmed the mechanism by adding activated carbon to invaded soils. Activated carbon absorbs organic compounds, including catechin. Native grass germination rates recovered substantially in treated soils, confirming that the suppression was chemical rather than competitive. After knapweed removal, catechin residue in the soil continues suppressing native grass recovery, creating a window where knapweed reestablishment from seed is more likely than native species return. Spotted knapweed matters because it introduced a chemical weapon from one continent into an ecosystem that had no evolutionary history of defending against it, and the soil chemistry makes recovery more difficult than removal. Japanese knotweed. Japanese knotweed is already known as one of the most destructive invasive plants in the United States. What is less widely understood is that it does not stop competing when it is removed. The soil keeps working for it. Rhinautria japonica produces allelopathic compounds from its root system, leaves, and leaf litter that suppress germination and early growth of competing native plants. Soils previously occupied by Japanese knotweed show reduced germination rates for native wildflowers and grasses, even after the knotweed has been physically removed. The chemical residue in the soil continues favoring reestablishment of knotweed from remaining root fragments over recovery of the native species that were displaced. The root system is why removal is so difficult to complete. Rhizomes extend 15 feet horizontally and 7 feet deep. A fragment the size of a fingernail left in disturbed soil regenerates into a full plant. Herbicide treatment requires multiple consecutive seasons because root stores enough energy to survive single applications, while the above-ground structure is being treated. The underground root continues releasing allelopathic compounds into surrounding soil, on riverbanks across New England, the Mid-Atlantic states, and the Pacific Northwest. The allelopathic suppression of competing vegetation removes native root structures that previously held streambank soil in place. Erosion rates on knotweed-colonized banks are documented as significantly higher than on native plant-covered banks. The soil chemistry creates conditions where the most stabilizing plants cannot establish, and knotweed does not replace the stabilizing function of the species it displaced. Japanese knotweed matters because its soil chemistry extends its competitive advantage well beyond its own physical presence, making recovery harder than removal. Sunflower. Most gardeners grow sunflowers without any awareness of what the plant is doing to the soil beneath it. Helianthus annus, the common sunflower, is native to North America and has been cultivated across the continent for over 3,000 years. It is grown commercially for oilseed across the Great Plains, used as a garden flower in millions of American yards, and planted as wildlife habitat across the country. It is also allelopathic. Sunflowers release phenolic acids and sesquiterpene compounds from their roots and through decomposing leaf litter and stems. These compounds suppress germination and early root development of competing plant species in surrounding soil. The allelopathic effect has been documented in agricultural research as a crop rotation problem. Fields that followed a sunflower crop showed reduced germination rates and reduced early growth in certain subsequent plantings without any other. Identifiable cause. Soil analysis in affected fields found elevated phenolic acid concentrations consistent with sunflower allelopathic residue. The compounds are water-soluble and move through soil with rainfall and irrigation. Breakdown rates depend on soil type, microbial activity, and temperature. In some field conditions, suppressive effects on subsequent plantings have been documented for more than one growing season after sunflower residue is incorporated. The same property that creates problems in rotation farming has attracted research interest for organic weed management. Studies have investigated whether deliberately incorporating sunflower residue into soil can suppress weed germination and reduce synthetic herbicide requirements. The plant's allelopathy may be an agricultural tool when managed intentionally rather than an accidental consequence of ordinary cropping. Sunflower matters because it is one of the most familiar and widely grown plants in American agriculture and gardens, and it has been altering soil chemistry around it without recognition. Eucalyptus. In California and along the coastal American Southwest, eucalyptus groves are known for a specific quality that visitors and ecologists both recognize immediately. Almost nothing else grows under them. Eucalyptus globulus, blue gum eucalyptus, was introduced to California from Australia beginning in the 1850s, initially for timber production and later for windbreaks and erosion control. The allelopathic properties were not documented or understood at the time of large-scale planting. By the time research had characterized the soil effects, eucalyptus groves covered significant portions of the California coast and East Bay Hills. Eucalyptus releases allelopathic compounds through 3 pathways simultaneously. Its leaves produce volatile terpenes that evaporate into the air around the tree and inhibit the germination of native species in the soil below. The leaf litter, which accumulates in deep layers because the leaves resist decomposition by most soil bacteria, leaches phenolic compounds into the soil as it slowly breaks down over years. The roots release additional allelopathic compounds directly. California native coastal scrub plants, native grasses and wildflowers that would naturally colonize the same habitat cannot establish under established eucalyptus stands. Purple loosestrife. Along wetlands and roadside ditches across the northern United States, this plant produces dense spikes of purple flowers in late summer that look striking from a distance. The wetland ecosystem underneath those flowers is being systematically dismantled. Lythrum salicaria arrived in North America from Europe in the early 1800s through ship ballast and herbal seed imports. It has spread across approximately 190 acres of wetland habitat in the United States and continues expanding northward and westward. It now occupies wetlands in nearly every state north of the Mason-Dixon line and across much of Canada. Purple loosestrife produces allelopathic compounds from its roots that suppress germination and establishment of native wetland vegetation, including cattails, bulrushes, and native sedges. Research has documented that the allelopathic effect operates independently of the physical shading and space competition that dense loosestrife stands also provide. Both mechanisms work simultaneously. The plant crowds out competitors physically while the root chemistry suppresses germination in the surrounding soil. Thanks for watching. If you made it this far, you already know what to do.